Okay. Very good afternoon, my dear friends. Welcome to today's session. So this is Dr.PADALA Tirupati, Assistant Professor, Department of Botany, Government Degree College for Women, Jagityal, along with our college, RBVR Women's College, Autonomous Hyderabad, SRR, Government Arts and Science College, Karimnagar, and TGCGTA, Telangana. We are all going to conduct this wonderful online Cpget coaching through online mode. Today's our eminent resource person, Dr.A.Madhuri Madam Garu, Assistant Professor, Department of Microbiology, Government Degree College for Women, Autonomous Begumpet, Hyderabad. Madam is going to explain today's one of the important area in molecular biology, that is transposans. So very, very important topic, my dear friends. Listen carefully. Madam, please start your session.
Thank you, sir. Hello, students. So we'll be today studying about two topics. One is transposons and the other one is mutations. And as we are preparing for entrance exams, and I just give you some important points in this topic, okay, where questions can be expected. Okay. So first, what are transposons? Another name for transposons is jumping gene. If possible, you can write notes, ma. You can take some points, running notes you can take. Transposons are called as jumping genes. Okay. Who has discovered? It is Barbara McClintock. Barbara McClintock has discovered these jumping genes. Okay. What do you mean by the term jumping genes? Yes, we understand these are genes. What are genes? Basically, genes are nothing but a sequence. Okay, a sequence of DNA. Right. When we are saying these sequences are jumping, they are moving from one place to another. They are called as transposons. Hope I am clear. Okay. Next. My voice is audible, ma'am? In which organism Barbara McClintock has identified these transposons is maize. One more possible bit. In which organism these are identified? It is maize. Okay. And this is Barbara McClintock. And it can be defined as a DNA sequence that is able to move or insert itself at a new location in the genome. Means it is moving, it is jumping from one place to another. What are the characters? They create novel genes since they cause mobilization, reshuffling, and rearrangements. Means just imagine a particular sequence is jumping from the genome from one place and going and sitting somewhere else. And the next site where it is entering, okay, that is randomly selected. It is not pre-decided, it is randomly selected. So it can jump to any other place in the genome. So definitely there will be changes in the functioning of the genes. In few cases, they can cause disease due to mutation. So there is every possibility of mutations here. Yes, mutations means this the genome functions will be altered. Gene functions will be altered. These are mobile genetic elements that may times carry an antimicrobial resistance gene. So not only they are moving from one place to another, they are also carrying some special genes. For example, antimicrobial resistance gene. Nowadays, there is more concern about spread of this antimicrobial resistance. Yes or no? You all should be interactive, ma. You all should be interactive. Okay. So when we are saying this antimicrobial resistance is spreading, means that gene is spreading from one organism to another organism and so on. Okay. So one of the parameter for antimicrobial resistance spread is also transposons. They code for some enzymes. Most important enzyme is transposase. Transposase helps in the mechanism or transposition. What is transposition? It is the movement of the transposon from one place to another. That process is called as transposition, and the enzyme is transposase. Write it down. Enzyme is transposase. Okay. Ikkada varaku ardhamaindamma, all of you. Next, we are going to discuss about types of transposable elements. So basic sequence or a basic type of transposon is insertion sequence or simple transposon. What is the name? insertion sequence, or in short form it is called as IS, insertion sequence or simple transposon. Okay, so these are short sequences. They are having the length of 768 base pairs to 5 kilo base pairs. And these are found in E. coli chromosome or bacteria. Insertion sequences are found in bacteria. And they are having, here I'll show you the structure. I hope this is clear, ma? This figure is clear for you? This is insertion sequence. Yes. No answer from anybody? Is this clear?
Yes, ma'am.
Okay, fine. So just to see the structure here, terminal inverted repeats and flanking direct repeats. There are two terms. Okay, terminal inverted repeats. So actually the sequence which is jumping is this much. From here to here, this sequence. Okay, so in this sequence, there is in terminal inverted repeat. Why we are calling terminal? Because this entire thing is sequence now. In this sequence, it is present at the ends. It is present at the ends. Okay, you don't consider this part. This is not part of insertion sequence. These two arrows are not part of insertion sequence. Only this arrow. Okay, and they are present at the ends. That is why we are calling it as terminal. And they are inverted repeats. One is in this direction and one is another is in this direction. Means they are in opposite directions. Yes or no? Are you able to understand? The sequence is in opposite direction. That is why we are calling it as terminal inverted repeat. Okay, now just we'll see the sequence. Here, this is inverted repeat now. So see the sequence on this strand. A, C, G, C, T, A. This is the sequence. Here, on this strand, A, C, G, C, T, A. It is present, but it is present on the opposite strand. Yes, it is present in inverted direction and on the opposite strand. So these are terminal inverted repeats because one sequence is moving towards this side and the another sequence is moving towards this side, right? So these are terminal inverted repeats. And what is present adjacent to this terminal inverted repeats? Flanking direct repeats. Flanking direct repeats. Now see the direction of flanking direct repeats, ma? It is in the same direction. Both the sequences are in the same direction. So here, see this T, G, C, A, that's all. Where is this direct repeat? T, G, C, A. Means both the sequences are in the same direction. That is direct repeat. But here one important point you have to understand is these direct repeats, they are not part of transposons. They are not part of transposons. From where they are coming? They are formed as a part of transposition mechanism. When a transposon moves from one place to another, it creates the formation of direct repeats. Am I clear? Am I clear here? What is insertion sequence? Insertion sequence is a simple transposon. It is having a particular sequence and at the ends it is having terminal inverted repeats. This much you have to remember. Am I clear, ma'am? Shall we move on to another part? Next part?
Yes,
Please
ma'am.
just okay. So just now I told you this direct repeat is formed as a process of transposition, right? So let us see how the sequence is moving. So suppose this is the transposon. Sorry. This is the sequence where the transposon has to come. Okay. So in this sequence, staggered cuts are made. One cut is at this site in this strand and the another cut at this site in this strand. See the sequence? It is same, but in the opposite direction. These two are complementary strands, right? So one cut here and the other cut here. Understood? So this kind of cut is called as staggered cut. This is important. This kind of cut at different sites in the DNA molecule is called as staggered cut. Staggered cut will form this type of molecules. See this? One strand is The sequence is more in this strand. Here, the sequence is more in this, right? So after staggered cut is done, transposon will come and sit here. This is the transposon. It has come and it is placed between these two strands, these two pieces. Okay, this piece has moved apart, this piece has moved apart, and between these two sequences, transposon is there. Okay, so what is happened after transposon has come? There is a gap here. There is a gap here. Okay, so that gap has to be filled by DNA pol 1, right? So when the gaps are filled, what they are forming? They are forming direct repeats. They are forming direct repeats. Ardhamaindamma? How the direct repeats are formed? In the target sequence, there will be staggered cuts made. As a result of staggered cut, we will be getting these sequences. After the staggered cuts, transposon will come and sit between these two pieces of DNA, and then the gaps formed are filled by DNA polymerase. As a result of gap filling, these two sequences are similar and they are present on either sides of the transposon. That is why they are called as flanking direct repeats. Flanking direct repeats. Hope I am clear. All of you?
Yes, ma'am.
Next, composite transposons. So we have seen simple transposons or insertion sequences. Next is composite transposons. Here, composite transposons means here. See this? Transposon will have inverted repeats. Just now we have studied now. What is a part of transposon? Inverted repeats or direct repeats? Yes. Transposon lo edamma. What is a part of transposon? Inverted repeats or direct repeats? Indulo, what is a part of transposon? Only inverted repeats is a part of transposon. Direct repeats are not part of transposon. Am I clear here? Without your interaction, it is difficult. Okay. So these inverted repeats are there here in this composite transposon, and between these inverted repeats, there is a genes for composite transposon will have insertion sequences on either side. Insertion sequences. Just now we have seen a simple transposon. This is simple transposon. This is simple transposon. Between these two simple transposons or insertion sequences, there will be another genes. There will be another genes. Two insertion sequences are lifting another sequence between them. Okay. So in this here, they can have gene for antibiotic resistance also. So these genes for transposition are present in insertion sequence only. So whatever sequence is present in the insertion sequence, okay, here it will code for the enzyme transposase, right? So similarly in composite transposon means it is having on either side what it is having, insertion sequences it is having. Insertion sequence means this is a small transposon, this is a small transposon. Two transposons together, they are lifting another part of segment, okay, with them. So that is composite transposon. Retrotransposons. Retro means RNA intermediate is formed. DNA is transcribed to RNA. Then RNA is then reverse transcribed to DNA. What is retrotransposon? Retrotransposon means first the DNA will convert to RNA, then RNA will again convert to DNA by reverse transcriptase enzyme. These retrotransposons are present in human genome. About 42% of the human genome is comprised of retrotransposons, and they can have this long terminal repeats, and they are called as line, that is long interspersed nuclear elements or short interspersed nuclear elements. These are the names of retrotransposons present in humans. Okay, LTR means long terminal repeats. They will have long terminal repeats, and they are called as lines and signs. Line means long interspersed nuclear elements, Sign means short interspersed nuclear elements. These are part of humans. Okay. So this DNA is converted to RNA and RNA back converted to DNA by reverse transcription. Next. Now we are going to study about how they are moving, mechanism of transposition. Just now we have seen. So this is the transposon. This is the target site. Okay. So this transposon will cut this target site and it will go and sit in this target site, right? And at the donor side, here transposon is missing. Ikkada transposon vellipoyaka, this is normal sequence, right? This is conservative transposition. Ante ఇక్కడ నుంచి వెళ్ళిన సీక్వెన్స్ వెళ్ళిపోయినప్పుడు ఇక్కడ ఆ సీక్వెన్స్ ఉండదు. దేర్ వోంట్ బి ఏ కాపీ ఆఫ్ ట్రాన్స్పోజాన్ హియర్. ఇక్కడ ట్రాన్స్పోజాన్ ఉండదు, కొత్త సైట్ లోకి వచ్చింది. దట్ ఈస్ కన్జర్వేటివ్. నెక్స్ట్ రెప్లికేటివ్ ట్రాన్స్పోజిషన్ మీన్స్ హియర్ ద దిస్ ట్రాన్స్పోజాన్ ఈస్ కాపీడ్. ఓకే, ఇక్కడ ట్రాన్స్పోజాన్ ఉంది, ఇది టార్గెట్ సైట్. సో ఇక్కడ నుంచి ఇది ఒక కాపీ ఫామ్ అయి అక్కడ వెళ్ళి కూర్చుంటుంది. సో డోనర్ సైట్ లో కూడా ట్రాన్స్పోజాన్ ఉంది, టార్గెట్ సైట్ లో కూడా ట్రాన్స్పోజాన్ ఉంది. Next, retrotransposition. Retrotransposition means this DNA is converted to RNA, transcribed to RNA, then reverse transcribed to DNA, and then it is going and sitting in the target site. Here also, donor site will also have transposon and target site will also have transposon. But the only difference between retrotransposition and replicative transposition. See, there are three types of transposition: conservative, replicative, and retrotransposition, right? Conservative means that transposon only go and sit in the new place. Replicative means it will form a copy and go and sit in the new. Retrotransposition means RNA intermediate will be there in the process of transposition. So that is retrotransposition. Next. So these are the transposons in different organisms. Bacteria, we can see insertion sequences, composite transposons. In maize, they are called as AC/DS systems. AC means activator, DS means dissociation. And Drosophila, we can see P elements, copia elements, FB elements. In humans, we call them as signs and lines. These are different terms you have to remember. You can get multiple choice questions based on this. Humans lo em untayi, P elements ఏ organism లో ఉంటాయి, copia elements ఎక్కడ కనిపెట్టారు. Okay, maize AC/DS system is seen in maize. Like retrotransposons can be seen in bacteria or not. There can be a question. Okay, right. So transposons you understood. I hope you understood. Right. Now we will solve some bits. Are you all ready? Yes, students. Now, transposable elements of bacteria are generally retrotransposons. True or false? I want all of you to interact fast. Yes, I need answer, ma. I may answer.
Yes,
Bacteria
ma'am.
transposons unnaya leva? They are present or not? Fast.
True.
Come up with answer. True or false?
True. True. True, ma'am.
Net lo check chesara? ఓన్ గా చెప్తున్నారా? It is false. Retrotransposons is seen in humans. The IS elements can be identified by the presence of. What is the IS element? What is the full form of IS element?
Insertion sequence.
Very nice, very nice. They are identified by the presence of. What it will have, ma? Antibiotic resistance gene untunda, endonuclease cleavage site untunda, inverted repeats untunda, what is there in this IS element?
Antibiotic resistant genes.
Anybody else? What is insertion sequence? It is the simple form of transposon which we have discussed in the starting. First type. ఏముంటుంది అందులో? Pujita.
Inverted repeat.
Very nice. Okay, we have discussed inverted repeat is a part of the transposon. Inverted repeat is a part of the transposon. Direct repeat is not part of transposon. Yes. So next answer is inverted repeat. Direct repeat in the IS elements are present. Direct repeats ఎక్కడ ఉంటాయమ్మా? Yes. Yes. We have discussed now direct repeats are formed as a result of transposition. They are not part of the transposition. Transposon, they are not part of the transposon, but they are formed during the movement of transposon. Okay, so ఎక్కడ ఉన్నాయి అవి రెండు వైపులా ఉన్నాయి ట్రాన్స్పోజాన్ కి రెండు వైపులా ఉన్నాయి సో ఆన్సర్ ఈస్ అప్స్ట్రీమ్ అండ్ డౌన్ స్ట్రీమ్ ఆఫ్ ద ట్రాన్స్పోజాన్ ఆర్ ఇన్వర్టెడ్ రిపీట్ నెక్స్ట్ డైరెక్ట్ రిపీట్ విత్ ఇన్ ద ఐఎస్ ఎలిమెంట్ హ్యాస్ అ లెంత్ ఆఫ్ డైరెక్ట్ రిపీట్ కి అరౌండ్ 5 to 15 base pairs ఉంటదమ్మా up to 15 base pairs length ఉంటుంది you have to remember this. Next, enzyme catalyzes the transposition of IS element is called as enzyme name antarama.
Transposase.
Very nice.
Transpose.
Yeah, transposase is the answer. Transposase restriction mechanism of IS element restricts the transposon and the target DNA in a combination of which of the following? ఈ transposase restriction mechanism ఎందులో మనకి transposition కాదు. See the conditions. Blunt end cut for transposon and sticky end cut for target DNA. Blunt end cut for both transposon and target DNA. Sticky end cut for transposon and blunt end cut for target DNA. Ikkada, what is the condition we have seen in the diagram is transposon ki blunt end cut undi. Okay, transposon ki emundamma blunt end cut undi and sticky end cut for target DNA. See the diagram. Transposon ki emundamma, this is blunt end cut, right? And this is staggered cut. అర్థమవుతుందా ట్రాన్స్పోజాన్ కి ఏ కట్ ఉండాలి బ్లంట్ ఎండ్ కట్ ఉండాలి టార్గెట్ సీక్వెన్స్ లో స్టాగర్డ్ ఎండ్ కట్ ఉండాలి క్లియర్ సో ఏ విల్ బి ద ఆన్సర్ విచ్ ఆఫ్ ద ఫాలోయింగ్ ఫంక్షన్స్ ఈస్ పర్ఫార్మ్డ్ బై ట్రాన్స్పోజేస్ అది మూమెంట్ ట్రాన్స్పోజిషన్ జంపింగ్ ఆఫ్ ద జీన్ ఆర్ మూమెంట్ ఆఫ్ ద ట్రాన్స్పోజాన్ లో అది హెల్ప్ చేస్తుంది హెల్ప్స్ ఇన్ ట్రాన్స్పోజిషన్ నెక్స్ట్ సెంట్రల్ బ్లాక్ ఆఫ్ ద కాంపోజిట్ ట్రాన్స్పోజాన్ ఎలిమెంట్ కన్సిస్ట్ ఆఫ్ ఎ జీన్ ఫర్ కాంపోజిట్ ట్రాన్స్పోజాన్ అంటున్నారు ఓకే దానిలో సెంటర్ లో ఏముంటుంది జీన్ ఫాస్ట్ Firdaus, what will be the central gene in composite transposon? రెండు చిన్న చిన్న ఇన్సర్షన్ సీక్వెన్సెస్ రెండు సింపుల్ ట్రాన్స్పోజాన్స్ కలిపి ఒక సీక్వెన్స్ ని ఎత్తుతాయి కదా ఒక దగ్గర నుంచి దే విల్ లిఫ్ట్ వన్ సీక్వెన్స్ సో ఇన్ దట్ సీక్వెన్స్ వాట్ ఆర్ ద పాసిబిలిటీ ఆఫ్ జీన్స్ ప్రెసెంట్ ఎస్ మోస్ట్ కామన్లీ దే విల్ హ్యావ్ యాంటీబయాటిక్ రెసిస్టెన్స్ జీన్స్ దే విల్ హ్యావ్ antibiotic resistance genes. Okay, next hybrid distances is caused by which of the transposable elements? Here you have to remember ma the hybrid distances and a process it is shown by p element. It is shown by p element and these p elements are present in Drosophila. Endulo untai p elements Drosophila. Am I clear here?
Yes.
Next, retrotransposons transpose through RNA intermediates. True or false?
True, ma'am.
Very nice. P strains of the Drosophila have active P elements. They'll have. True. LTR
Yes, ma'am.
retrotransposons are abundantly found in. ఈ LTR retrotransposons ఎందులో ఉంటాయంటే దే ఆర్ ఇది ఆన్సర్ ఫౌండ్ ఇన్ లోవర్ యూకారియోట్స్ లైక్ ఈస్ట్ అండ్ డ్రోసోఫిలా LTR retrotransposons లోవర్ యూకారియోట్స్ లో ఉంటాయి ఓకే లైక్ ఈస్ట్ అండ్ డ్రోసోఫిలా సో ఆన్సర్ ఏమొస్తుందమ్మా ఆన్సర్ విల్ బి ఇక్కడ ఇది ఆప్షన్స్ ఈ ఆప్షన్స్ లో ఆన్సర్ చెప్పండి ఎస్ వినపడట్లేదమ్మా
Yeast, ma'am.
ఈస్ట్ గుడ్ లైన్ అండ్ సైన్ ఆర్ ఎగ్జాంపుల్స్ ఆఫ్ ఇందాక చూసాం కదా
LTR
Non-
retro.
retrotransposons they are present in vertebrates humans లో ఉంటాయని చెప్పాను కదా ఇవి ఓకే దే ఆర్ ఎగ్జాంపుల్స్ ఆఫ్ నాన్ LTR retrotransposons ఇక్కడ డిస్జెన్సీస్ occur in every cell of డ్రోజోఫిలా. ఇది చెక్ చేయాలమ్మా. ఆ ప్రాసెస్ ఎవ్రీ సెల్ లో అవుతుందా లేదా అనేది. Key elements are responsible. Just check this question. Okay. Disgenesis ప్రతి సెల్ లో అవుతుందా లేదా. Okay. Answer is false. ప్రతి సెల్ లో కాదు. Okay, it won't occur in every cell. So with this we have completed bits for transposons. Next we are moving on to mutation. Okay. Still we have 10 more minutes. I'll try to complete this. Shall we start? Generally, you will have you are having basic idea about mutations, right? Based on this, whatever books you are having, that bits are there, you can solve at home. I'll just give you the basic concept. Mutation means, what is mutation?
Sudden heritable.
It is a change in the genes sequence which can be heritable to next generation, right? It is a permanent alteration of the sequence. Next. So what is the cause? Why mutations are occurring? It can be due to mistakes in replication or other environmental factors are also responsible. Okay, about 1 out of every, just count this number, 10 power 7 times a mistake occurs during copying. So inni saalu copy form ainappudu there is a possibility of one copy carrying a wrong sequence. Okay, what is the environmental factors? There can be Tirupati sir. So how is your preparation, all of you?
Okay, my dear friends, due to power interruption, Madam unable to share her videos, her slides. So our session is going to end, my dear friends. And one more, today we have one more session at sharp 5 p.m. So my dear friends, all of you join sharp 5 p.m. Okay, my dear friends. Right.